Today we're going to talk about phototransduction in the rod cells of the retina, starting with the eye. The many different parts of the eye are the iris, the pupil, the cornea, the ciliary muscle, the fovea, which is where vision is sharpest, the lens, the blind spot, the retina or tunic layer, which is the innermost layer of the eye and what contains rods and cones, and the optic nerve. Now, into the meaty stuff. Okay, say this is one photon of light. This photon is going to travel into the eye and hit the retina, most likely near the fovea. The retina has rod cells which are very sensitive, and they allow us to see in dark lighting such as moonlight or twilight, and cones which allow color sensitivity. Now we're going to take a look at what goes on inside the retina. Here, inside the rod cell, you have a visual pigment called rhodopsin. It's made up of an opsin protein molecule and cis-retinol. When one photon of light strikes rhodopsin, the cis-retinol changes into trans-retinol, which removes the attraction between trans-retinol and the opsin protein, exposing the binding site. Let's put this aside for a while and build transducin. Transducin is made up of 3 subunits, one of them being GDP, or guanosine triphosphate. There's also phosphodiesterase, or PDE, which converts cyclic GMP, cGMP, to simply GMP. But it cannot do that with the 2 alpha subunits attached to it. But we'll get to that later. The opsin with the exposed binding site moves towards transducin. Since the opsin has an exposed binding site, it can catalyze a reaction. The GDP subunit adds a phosphate and turns into GTP. That new subunit leaves the other 2 to join one of the alpha subunits of the PDE, which helps the PDE lose one of its inactivating alpha subunits, which makes it half happy. Now, in order for the PDE to complete its intended purpose, that whole reaction starting with the photon and ending with the transformation of GDP to GTP must happen again so the PDE can lose its second alpha subunit. Once the PDE becomes isolated, it can fulfill its intended purpose of transforming cGMP to GMP, which will close cGMP-gated sodium channels, essentially giving us vision.